Yes, hello, hi, can you hear me?
Yes, yes, I can. How are you, Daniel?
Fine, thank you. How about
Mm.
you?
I'm good, doing well. Thank you. Yeah, we will start in a moment. I think it is
Okay,
taking,
okay,
yeah,
just fine, just let me know.
maybe in 5 minutes we will start.
Sure.
Very good afternoon, Professor Daniel from India. Yes, can you hear me?
Yes, I can
Yeah.
hear. Yes, good afternoon.
Good afternoon. On behalf of the Department of Physics, I welcome you to this university for this special physical activity session talk. Thank you for accepting our invitation. Before we start, actually, as it is a customary, you introduce the invited speaker to the audience. Professor Daniel, we have included a neighboring institute also for today's talk because to maximize the benefit that we can extract from your talk. So we have also included audience from neighboring institutes and also from neighboring states like Manipur and also from Dibrugarh is within our state only. So we have maybe 4 or 5 institutes who have joined this talk online. So let
Okay.
me introduce Professor Daniel before the audience before he start his talk. Uh, Professor Daniel has completed his diploma in chemistry from the Department of Chemistry, Swiss Federal Institute of Technology, Zurich, in 1992. Then he completed his short-term research stay at the University of Tokyo in 1996 to November 1996, from August 96 to November 96 only. And then he obtained his Doctor of Natural Science from Swiss Federal Institute of Technology, Zurich in 1998. And 2003 to 2004, he did his postgraduate studies to obtain the qualification as Masters of Advanced Studies in Intellectual Property at the Swiss Federal Institute of Technology, Zurich. So as far as his work history is concerned, he has quite a long list of work history. From 1998 to 2000, he is a postdoctoral fellow as a JSPS postdoctoral fellow at the Department of Applied Chemistry, Keio University. He is a Science and Technology Agency Research Fellow at the Kanagawa Academic Science and Technology, CAST, under Professor Suzuki. From 2002 to 2000, October 22, he is a researcher at the Kanagawa Academic Science and Technology. From November 2002 to September 2003, he was a senior research scientist and deputy of the director at the Center for Chemical Sensors of the Swiss Federal Institute of Technology in Zurich, Switzerland. He is a patent attorney in the Patent and Scientific Information Department of Headquarters of SIBA Specialties Chemicals in Basel, Switzerland. From 2006 to 2007, he was a special research associate professor at the Faculty of Science and Technology, Keio University. And he was an associate professor in between 2007 to 2009 at the Keio University in the same department. He was a part-time researcher at the Kanagawa Academic Science and Technology from 2009 to 2014. And he was an associate professor at the Department of Applied Chemistry. I think it is from 2009 to 2014, he was an associate professor at that university. From 2014 to till date, he is a professor, full-time professor at the Applied Chemistry Department, Keio University. So besides this, he is an active member in various important societies related to chemistry and other bodies. To name a few, like American Chemical Society, Chemical Society of Japan, Japan Society of Analytical Chemistry, and then the Japan Analytical Instrument Manufacturer Association, Micromachine Center Beans Knowledge Database Committee member, and JIMA JSEC PITCON Symposium Committee member. He is a National Institute of Education Policy Research external member of the Committee for the Promotion of Globalized University Education. He is the RSC Tokyo International Conference Organizational Committee member. He is a Fellow of the Royal Society of Chemistry, FRSC. From April 2017 to present, he is a member of the Permanent Steering Committee of Europort, and he was the editor-in-chief of the journal Sensor and Actuator B Chemical between 2018 and 20. Currently, he is a co-editor-in-chief of the same journal. And from April 2019 to present, he is an international advisory editorial board member of the Journal of Indonesian Chemical Society, Chemical Society. And from February 2020 to December 2022, International Advisory Board member of Analytical and Bioanalytical Chemistry. And from 2021, January 2021 to present, Advisory Board member of the Analytical Chemistry, ACS Sensor, American Chemical Society. And from January 2023 to present, he is the Advisory Board member of ACS Sensors. So with this, I would like to request Professor Daniel to start his presentations. So Daniel, the stage is all yours.
Thank you very much for your kind introduction. So let me first try to share my screen because actually it's my first time to use Google Meet. So I will try now and see what happens. So I hope that you can.
Yes, we can see your presentation. Can you make it in a
Okay.
slide mode? No.
Okay, just a moment. Can you also see my pointer now moving on the screen?
Yes, we can see. We can see your presentation.
Okay,
Yes.
okay.
The
So
pointer
just to
is
let
there.
you know, I don't see you at this time. I don't see anything. I only see my PowerPoint slides. Okay. So first of all, so thank you very much, as I just mentioned, for the kind introduction, and thank you very much for giving me the opportunity to give an overview of our work that we have done and are doing. Of course, within the time of approximately 50 minutes, I will not be able to show you everything that we have done. But yeah, I would like to focus, as you have eventually seen in my abstract, I would like to focus on two different topic groups. The first one is about how to make analysis as simple as possible. And the second one is some of our more recent activities. And actually, the final one is also where Professor Pabitra Nath contributed a lot when he stayed in our laboratory here in Japan during the last year. So, and you are all from physics, so I must apologize. So some of my content, well, all of my content is related to analytical chemistry. So yeah, I hope that you can follow what it's not very heavy, difficult chemistry. So I try to make it as simple as possible. So our group, starting in 2008, our group has worked on the development of what we call paper-based analytical devices or parts. And the reason is, yeah, first I'd like to give an answer. So what's the motivation for using paper? So I hope that the slide has changed to the next one. Yeah. So especially when we talk about on-site analysis, especially medical diagnostics, but this is not limited also for environmental analysis or looking at the safety of food, there's a continuously growing need for on-site analysis. That means analysis that has to be done outside of the laboratory. And therefore, so high-tech analytical instruments are not an option because, first of all, they're too expensive. They can only be operated by experts. And of course, they are not portable and they cannot be taken out of the lab and they require electric power and continuous reagent supply. So now in the field of analytical chemistry for several years or even decades, there is a trend to try to develop more simple and low-cost analytical devices. And so why paper? Well, as probably all of you know, so paper has been used in analytical chemistry actually for centuries. So here, this is probably one of the oldest analytical systems. It started with the litmus paper in the 17th century. But nowadays we have many commercially available systems where we have colorimetric pH indicators immobilized on a piece of paper. So we can very easily use that and we can make very simple pH analysis. Then for medical diagnostics in the 1950s, the so-called urine dipsticks have been introduced. So these are pieces of plastic which has pieces of paper stick to it and where some colorimetric reactions take place so that we get a rough overview of our health status by looking, for example, here at several target analytes at the same time. And then in the 1980s, another very important type of paper-based analytical devices was introduced. These are the so-called lateral flow immunoassays, or LFIAs. And due to unfortunate happening of the global COVID-19 pandemic, now everyone knows about this system and has probably used these for the rapid COVID antigen test. So what are the common features? These all include paper as a material, and the paper has been loaded with the reagents that are necessary for the analytical test. They're normally single use, and they give us a semi-quantitative or qualitative results. Of course, they have advantages of being relatively low cost. They're very easy to use. And once you have used them, you can dispose them easily. So these are now established analytical platforms made of paper. And then in 2007, a very famous chemist from Harvard University in the United States published a paper in the journal Angewandte Chemie, and he introduced the concept of so-called microfluidic paper-based analytic devices, micropads. At first, nothing much was new. It's still paper platform with reagents on it. But so I mentioned several advantages of using paper already. Of course, paper is low cost. It is very lightweight. You can easily dispose of. But one very important aspect that I've not mentioned before, it's a porous material. So any liquid can flow in paper autonomously without any pumps. So based on capillary flow. And actually, the lateral flow tests for the COVID-19 rapid antigen test, it's based on this capillary flow. So that's a very important characteristic. And also we have a large surface area where we can immobilize reagents. So what Professor Whitesides did, they introduced this microfluidic structure. So having these patterns, paper substrate with flow channels, we can do several tests with relatively small volumes, and we can do multi-step assays. That is not possible with a simple pH test paper. That's a one-step reaction. And if you make this relatively small, we can save on the sample volume, and this reduces the cost of the reagents that are consumed. So we can make it more low cost. Actually, the World Health Organization has in 2004 created this concept of assured. So that means these are the requirements for point-of-care testing devices. So what does it mean? It means if we want to use an analytical system at the point of care, that means outside of the lab or in very remote settings, then these analytical systems should fulfill these criteria, abbreviated as assured, which stands for affordable, sensitive, specific, user-friendly, rapid, and robust equipment, free and delivered. And the already commercially available paper-based analytical devices and those that have been developed since the first micropad introduced by Whitesides Group about more than 13 years ago, they already fulfill many of these criteria, not all of them, but especially when it comes to user friendliness, or when we want to get analytical results without using technical equipment, then there's still room for improvement. And in fact, there's also, besides the WHO, there's another very important legal feature that's issued by the FDA, Food and Drug Administration of the United States, which basically states which kind of analytical tests can be used or are acceptable for use by ordinary people, like not experts, not medical doctors, not clinicians. And these are the so-called CLIA-waived tests. By the way, this COVID-19 rapid antigen test was as such a CLIA-waived test. That means it can be used by ordinary people. And it says that in order to be able to be used, it must be very simple. So basically the method must be so simple and accurate as to render the likelihood of erroneous results by the user negligible. So in other words, so operational simplicity is extremely important. So there already are commercially available paper-based analytical devices. So I mentioned the urine dipstick test. So you can dip that into a urine sample. Then according to the manufacturer, you have to wait for 60 seconds or for 1 minute. And then you compare with a color chart and get a rough estimation, in this example, of the glucose concentration. And then many researchers have developed very nice and excellent so-called micropads, these microfluidic paper-based analytic devices, for example, where you get information about the target analyte concentration by measuring the time. You add a sample here and you measure the time until a colored spot appears here. Or you count the number of colored lines, as in this case. Of course, these are fine for semi-quantitative naked eye readout, but still, they have some issues and they're not necessarily suitable for untrained users. So, for example, here, if you miss the 60-second timing, the results will no longer be reliable. The color that you see here depends very much on how bright your room or the environment is. So these are some issues. And we have started working on these paper-based devices actually also in 2008, just 1 year after the first paper has been published. And but of course, I cannot introduce all of the work here. So I would like to introduce here a few representative examples showing you what we try to do to make the analysis for the user as simple as possible, while at the same time making the readout or how to collect the signal also as reliable and as simple as possible, ideally without using any instruments. So that the systems can be used by ordinary people, so untrained end users. And we refer to this as sample in, answer out. So you add your sample and then you get the answer. I'd like to shortly introduce 4 different types inspired by very everyday situations. First of all, for example, read the result in the form of text. Or read the result like we read temperature from an analog thermometer, where we look at the length of a mercury part or like a colored alcohol, but this time for a different, not for temperature. Or we use QR codes for signaling for this traffic light system. So let me start with the text. One example of how we can use text. And actually, in this case, this we have already published 4 years ago in ACS Sensors. Here we collaborated with a company and they were interested in having an intuitive test for measuring oxidative stress. Oxidative stress can be measured, for example, by detecting DNA decomposition products in human urine. And there already is a commercialized system, and that's very similar to the COVID-19 rapid antigen test. It's such a paper-based lateral flow immunoassay. But in contrast to the COVID-19 antigen test, there's something different here. So I cannot, for time reasons, I cannot go too much into the details. But we have to detect a small molecular weight compound, which is the DNA decomposition product. And when we do so, we have an opposite situation from the COVID-19 test. That means in the positive sample, so when we have a high concentration of this DNA decomposition product in our urine, the test line is disappearing. So if you have done a COVID test, you know that if you are COVID positive, the line is appearing. So that's a very intuitive way. But for small molecular weight compounds, this assay actually goes the other way around. So the line is disappearing. And so this can be very confusing for ordinary users. So what we have done in a nutshell, so we have converted this so-called signal turn-off system, where the line is getting thinner and thinner and is disappearing, in a signal turn-on, where we can read the approximate concentration of the DNA decomposition product in the form of text. That means if there's nothing in your urine, you just see TR, which stands for trace. If you have a very high concentration, then you can read this high. And just on the side, so this is the compound we are detecting here. It's called 8-OHDG. In short, it's 8-hydroxy-2-deoxyguanosine. So this is a urinary oxidative stress biomarker generated by DNA damage, and it can serve the purpose of assessing the risk for cancer due to lifestyle-related diseases. What we have done, so here there's nothing revolutionary new. What we have just done is we used the known reagents. So this is a so-called immunoassay. So we work with antibodies and antigens, but we rearrange them in a specific way on paper, and we create where we mix two analytes. One of them we refer to as a text antibody. Why? Because we deposit it in the form of text. So here's the character LO. And the second one, this green compound here, we refer to as the mask antigen, which we deposit around it. And we do all of this by inkjet printing on paper. So in the negative sample, you only see the mask. So all of these appears as red. But if you have 8-OHDG in your urine, the mask is getting thinner. At the same time, the text is getting stronger and we can read this. So that's the very simple working process. As just mentioned, so the device looks like the COVID-19 antigen test. We deposit the urine and we fabricate this by inkjet printing. So this is a standard office inkjet printer that we use. And in negative, you see this mask pattern. When it's positive, you can create whatever pattern you like by inkjet. You can have numbers, you can have letters, or here the analyte. And by controlling the amount of these two reagents on this spot, we can control the concentration of this DNA damage biomarker when our text becomes readable. And we do this by inkjet printing. So we modulate the ratio between the mask antigen and the text antibody by inkjet printing. And that means that the DNA damage biomarker in urine, the more of these text symbols will become readable. So it's like reading a book in the most simple way. So we can get semi-quantitative information, whether your concentration is very high, whether it's low, or whether it's basically nothing, so it's trace. Of course, I will go through this very quickly. So we tested this with real samples in the hospital, and so we measured the absolute values, the absolute levels of this DNA damage biomarker using a hospital instrument, and we compared with our very simple system, and we used this color coding. So ideally, these two columns should show the same color. You will see that it's not perfect. So we have some outliers here. For example, we detected too low concentration. Here we detected too high concentration compared to the hospital value. But in generally speaking, so for this very simple method, we were very happy with this result. And if you compare that with the commercially available test that has been on the market so far, the detection limit in that case was about 1 microgram per milliliter, whereas with our system, we have been able to identify concentrations down to 3 nanogram per milliliter of this DNA damage biomarker, but without using any equipment. So if you use an electrochemical analyzer, then there are, of course, systems in the literature which are more sensitive. But yeah, so this is one example just by rearranging known chemical reagents, so the antibody and the antigen, so it's an immunoassay, we could simplify the detection principle and actually improve the detection limit. The next simple example that I would like to show you is where we use this inspired by the analog thermometer, where we detect by naked eye the length of a colored zone on paper. And the analyte that we have chosen here, again, this we have done in collaboration with a research group in Taiwan and a hospital in Taiwan. And we target another analyte in human urine, and that is the so-called urinary albumin index. Albumin is a protein. It's the smallest molecular weight protein that you find in urine. Well, actually, you should not find it in urine if you are fine. However, if your kidney starts to fail, so if you have a kidney disease, the level of this protein in urine will start to increase. However, the concentration in urine itself is not a very reliable parameter because concentration of all compounds in urine depend very much on how much you drink, what you drink, and therefore in clinical laboratories, physicians or clinicians use creatinine, which is another compound that is excreted in our urine, and this is mostly excreted at constant levels. So they use it as a kind of internal standard for normalizing the amount of albumin. And this parameter is called the urinary albumin index. So it's simply the ratio between creatinine and albumin. And doctors are interested to know to easily distinguish these three states, like the normal state, the so-called microalbuminuria, when there is still good chance for therapy. If you have advanced to macroalbuminuria, then your kidney is already heavily damaged. So this should be detected as early as possible. So what we have done here, we have developed a paper-based device which directly shows us graphically this urinary albumin index without using any cameras, without using any software, any computer. They've simply done this. So this is a scheme of the device. You have two channels on the device, one for creatinine, the other for albumin. You dip it into the urine sample and you look at the length where the color has changed. So like an old analog thermometer, like when the temperature increases, for example, this color line here increases, but here it's not the temperature, it's the creatinine concentration. When it increases, the yellow part becomes longer, the same for albumin. And then we just draw a straight line connecting the points where the color has changed, and we look at where the intercept of this drawn line comes. So if the intercept here is in the blue range, that means you are fine, you're healthy, there's no problem with your kidney. If the intercept gets into the red range, then this becomes a problem. And this intercept does not depend on the absolute concentrations of the two target analytes of creatinine and albumin, but only on the ratio of these two. What is the principle behind this? So here is a part. The part is well, physical chemistry or chemistry. Actually, we make use of the fact for albumin that proteins are very strongly adsorbed to the surface of paper, and since the proteins, when they flow, when they wick through the paper channel, they are gradually absorbed. That means we deplete the sample. So the longer the sample flows, the less protein is contained in the sample. And we deposit an indicator which is sensitive to protein. As long as we have protein in our sample, the protein is gradually absorbed and it will change the color of the indicator from yellow to blue in this case. And once there's no more protein, there will be no longer color change. So that means the higher the protein concentration, the longer the section of this blue channel and the shorter the yellow one. For creatinine, for time reasons, I will skip the details. It's a similar process, but we use a different chemical concept here. Again, we adsorb the analyte creatinine partially on the paper and on this reagent that has been immobilized on the paper to get this length, concentration-dependent change of the yellow color. If we do so, that's how you experimentally observe. Of course, you can quantify that the higher the albumin concentration, the longer the distance or the length of this color change distance, the same for creatinine. And so this is quantitatively evaluated, but as I mentioned, so we want to do that visually. So here we looked if we do that with these have been artificial urine samples. So this albumin index going from left to right is again, I used a color coding. By the way, here in this table, you will find the value of the absolute value of the albumin index measured on a clinical large scale high-tech clinical analyzer in the hospital. Again, this we have color coded. And as you can see here, with our very simple system, we have been able to identify all of these cases. So we have the healthy ones where there's no problem here. So the intercept of this drawn line is in the blue area, in the safe area. And then, for example, we have these three patients with very progressed kidney issues. So you can see that the intercept is here in the totally different range. So that is another demonstration where we use just our naked eye. And here there's no new chemistry involved. It's just the idea of how you arrange the reagents on a piece of paper, how you make use of the interaction between your analyte and the paper, or your analyte and the reagent, and then you can create such a system. This we have also published in ACS Sensors approximately 4 years ago. The next example, this has not gone to clinical testing so far. Well, you are certainly all familiar with QR codes. They are now found everywhere, like whether it's on boarding, online boarding passes for getting on the airplane, whether it's on giving an URL to a website. So here we tried to use a QR code for a simple analytical device made of paper. And this is, so far, as I just mentioned, it's no clinical application yet. So we just did a proof of concept. So what did we do? First of all, we heavily work with inkjet printing. So that's the workhorse for our device fabrication. Again, we use a standard office inkjet printer. We remove the original inks and we replace the original inks with chemical reagents. So therefore, we can print a QR code, but not with a standard color ink, but with a reagent. In this case, we use the metal ion indicator. So if we print a QR code with that, this compound specifically is responsive to copper, Cu2+. We have this red color when there's no copper. If we dip it or if we have the presence of copper, then it changes to this bluish color. So what did we do then? We masked this QR code. So when we have no analyte, when we have no copper, we want this to become unrecognizable. Then we deposited again by inkjet printing another dye around this QR code, so this is no longer readable because of the contrast. But when we add our sample, it becomes readable. So we have a turn-on situation. So this would be not readable, and this QR code gives you a message on your smartphone. And then we combine these with this like thermometer-like system, the analog thermometer-like system. In other words, you deposit your sample, and the higher the concentration of your analyte, in this case, the higher the concentration of copper, the more of these different QR codes will become readable. So if you have a very high copper concentration, like here, all 3 QR codes become readable. And that will tell you your smartphone will tell you the approximate concentration. And yeah, this is how we actually make this. Again, I will skip the details here. It's all done by printing. We use filter paper. We pattern these to have a flow channel. Again, by printing, we print all the reagents. And here we did the test. So when there's no copper, none of these QR codes is readable. When we increase the concentration of copper in this is an artificial sample. So it's again no clinical meaning. The number of readable QR codes increases. So this is another example of how just using a piece of paper, very well-known chemistry, but yeah, arranging the reagents in a different way, we can create a new analytical system that is very easy to use. So a fourth example that I'd like to show you here shortly, again, is inspired by something that is around us in our daily life, and these are traffic lights. So here we call this a traffic light paper-based device. And why are we interested in having this traffic light device? Well, it's the highest level of intuitiveness that I think you can get out of a piece of paper. Let me explain that a little bit more. So now, for example, I have shown you this text readout of a lateral flow immunoassay. And I have shown you this urinary albumin index system. For example, here up to where is the yellow color, and up and here up to where is the blue color, and then draw this line. So this system work, these two systems work, as I she has to make a decision. So some people might say, okay, like let's use this example, might say, okay, I can read the high here, not only the medium, but actually we just want the medium to be readable. Whereas some people might not be able to recognize the high here. So there is some difference from depending on user's eyesight and so on. The same applies for this system. So the user has to judge, okay, where is the end of this yellow colored line? Where does the blue start? So there's still, this requires user interpretation. There is a risk of readout error. And so therefore it's very, or in other case, like if you do the COVID test, there is basically no readout error because either you see the line or you don't see it, but this is very qualitative. It just gives you a yes or no answer. So then we thought if we can have this traffic light system that everyone around the world knows, like green if it's safe, well, yellow if you should take care, red if it's in danger, that would be a good system. So how did we realize that on pieces of paper? Here comes the chemistry that we use. So what we are doing is inside paper, we are controlling flow of liquids. And we used this compound. So this compound, since I assume most of you are non-chemists, just very shortly, this compound is hydrophobic. It means if we deposit this compound in a flow channel in a piece of paper, it will block the flow of an aqueous liquid, like urine or blood, for example. Here we used artificial urine, so it will block that. However, this compound will react with hydrogen peroxide, H2O2, and will be decomposed into hydrophilic compounds. So we call this a chemical valve. So we have blocked flow when there's no hydrogen peroxide, and we have free flow when there's hydrogen peroxide. So here we are not interested in directly measuring hydrogen peroxide. However, in the field of biosensors, there are many enzymes belonging to the oxidase family, and like glucose oxidase, cholesterol oxidase, and so on. So what do these enzymes do? They convert important biomarkers, for example, glucose or cholesterol, into an oxidized metabolite, and at the same time, they create hydrogen peroxide. So we can use such a chemical valve, which is sensitive to hydrogen peroxide, for detecting very important biomarkers. In this example, we used glucose by combining it with glucose oxidase. How did we do that? So this is a very schematic figure. So these are 7 layers of paper. I will show you the actual device in a moment. These are 7 layers of paper that we have patterned and modified. Actually, I cannot show you all the details because this would take too much time. So we did a lot of modeling and calculations on length of this channel, what flow speed we have to do here, achieve here, and so on. And but we place these. Sorry, I will just go back one slide. We place this hydrophobic material on this paper, and we call it a hydrophobic wall. So we place different amounts to block the flow. And we also use food dye. So this here is a food dye, a green, a yellow, and a red food dye. This is no chemical indicator, not chemically reactive. So we simply use food dye. So what happens now? If you have no low hydrogen peroxide in the sample, for example, no glucose in the enzymatic reaction will give us no hydrogen peroxide, then our liquid, our urine or artificial urine, cannot flow to the red channel, cannot flow to the yellow channel, but only to the blue channel. So we will get the green channel. So we will get a green signal. If you have very high hydrogen peroxide concentration or schematic figures of these 7 paper layers. So these are 7 layers of paper, and we fold them, stack them together. Actually, here you can see the greenish. It looks more blue, but yeah, it's a greenish, yellow, and red food dyes. On the top, we put our enzyme, and these are the channels. And here in these spots, we deposited this hydrophobic compound, which blocks the flow. And then we fold it, put everything together, apply our artificial urine sample, and observe the backside and get this signal. And this is the result. I jump directly to the results, how this looks. This has been done in artificial urine samples. So please look at this figure here first. So when you have no glucose in your urine, that means no risk of being diabetic, then you only see the green spot. As soon as your glucose concentration in urine starts to increase, as a first warning, you will be able to observe the greenish and the yellow spot. That means, okay, your glucose levels are starting to increase. If they increase further, you will only see the yellow spot. So this is the next level of warning the patient. In this case now, we believe is totally independent of any user interpretation, because you don't have to judge a faint color. You don't have to judge whether you can read a text or not. You don't have to judge how long is the length of a color changing line. You simply look at this, like you do at a traffic light. And by doing so, of course, this is not quantitative. We call it semi-quantitative, but you can still very easily distinguish between 5 different levels of glucose in your urine. And compared to the commercially available urine dipstick, where you have to judge faint color changes, this is much more obvious and much more intuitive. Okay, so with that, I'd like to sum up just this very, very first part. So before coming to a little bit different topic, I hope that I have shown you that with some piece of paper, we can do actually reliable analysis in real clinical samples. Not yet, we have not demonstrated all the applications in the clinic yet, but yeah, these have been examples. And the paper is for us a very important thing. So it's very important material for us. So one point, of course, as you probably understand now, we heavily depend on this capillary force-driven liquid transport. Without that, we could not observe all of that, what I have shown you right now. It serves as a container or a surface where we can pre-store also the combination of paper and printing. This gives us a lot of freedom of how we can design. It also makes the devices reproducible, because as reproducible as you can print a photograph with an inkjet printer, you can print a device. And we think, as we have shown in a review, so this review has already been published 7 years ago, we think that these paper-based devices will hopefully really become more useful tools for general health state monitoring. And also we'll find some niche applications even in high-tech hospitals. So that has been the part one. Here I really focused on simplicity. And now for the last about approximately 15 minutes, 13 to 15 minutes, I'd like to show you something different. We still stay on paper, but here it's not as simple. Well, it's still simple compared to a lab instrument, but here we focus especially on sensitivity. So that means we want to detect very low concentrations. And here we use a tool that has become very popular in bioassays, but just during, I would say, the last, well, it's probably less than the last 7 or maximum 8 years. And this is the so-called CRISPR-Cas system. So I don't know whether you have heard about it. Actually, a Nobel Prize in chemistry has been awarded for the detection of this system, but for a different reason, not for analytical chemistry. It can be used for gene editing, but we don't use it for gene editing. So in our case, it's totally different application. Let me shortly introduce what this is doing. So Cas is an enzyme, and CRISPR stands for clustered regularly interspaced short palindromic repeats. So that's basically it's originally isolated from immune defense system of some bacteria. But so we use so-called Cas12a enzyme. What does it do? It has the enzyme, and it's combined with the so-called CRISPR RNA, this green line here, cRNA. And it can detect a complementary DNA sequence, double-stranded DNA. So this yellow part here. So here the red part and the green part are complementary. So it recognizes the complementary double-stranded DNA sequence. When doing so, this enzyme is activated. It cleaves this double-stranded DNA at several places. So it chops up the DNA. But that's not what the analytical chemists are interested in. What we are interested in is the so-called transcleavage. It actually indiscriminately cuts nearly any single-stranded DNA piece that is in its neighborhood. And this can be used for analytical chemistry purposes. In other words, we use this as a recognition system. So if we have this double-stranded DNA, we activate this enzyme which will cut this single-stranded DNA, and this we can convert into a signal. So we use a model where we have no signal, no fluorescence when this single-stranded DNA is intact, but when it is chopped up, we turn on the fluorescence. And this has now been used in paper. Actually, the first report on paper was just about 3 years ago, published in Nature Biotechnology, where this is a foldable paper device and where they used this. So actually, these CRISPR-Cas systems can be used. They are compatible with paper platforms. And the very, very first application involving paper actually was in Science. This is CRISPR and the CRISPR RNA, because RNA is inherently unstable, but they can be pre-deposited on paper, because these researchers have not looked into that. They added the reagents and immediately performed the reagent. But we wanted to look, can we make a system that we can actually use at point of care, and therefore it has to be storable. So just very shortly, first we did a basic evaluation of the stability of this system on the paper substrate. And we did this because of time reasons, because I'm running out of time. I don't want to go too much into details. So we looked at how can we dry these reagents? So do we need freeze drying? Do we just let it stand and dry? Should we use an oven drying? How do we have to store it? Can we add stabilizers? And we did this. We just very recently, actually just this summer, published this very simple work. And we just used this paper spot where we detect a fluorescent signal. And if we can detect the fluorescent signal after storage, then it means the system works, so it's fine. This has no analytical meaning. This is just for evaluation. So we did this very simple test. I skipped the experimental details here, and I just would like to show you the results here. So we have shown, in this case, it is just 1 month, but actually we have monitored this for up to 90 days, for up to 3 months. This is a very simple double-stranded DNA sensor on paper. We can store it. We can store this CRISPR-Cas system on paper. For here, it's shown for up to 1 month, but as I mentioned, we have evaluated for up to 3 months. So stored in freezer minus 20 degrees, this system works. So the higher the concentration of DNA in the sample, the higher the signal will be. Actually, just to compare, if you store this system in a microtube and just freeze it, it will not be, it works on day 0. That means when the solution is fresh, but as you can see, you get a strong decrease. So actually, the paper here, the paper that we use, has a stabilizing influence on this system. So this has been very good news. And since we found that out, we wanted to show a real application, and that will be the last topic. And that was actually the work that also Professor Pabitala Nath has been involved here with us. Well, except, of course, he has also been involved in other works, but this is the work that I'd like to shortly introduce. So here we are not looking at analyzing nucleic acids, not analyzing DNA, but we perform an immunoassay. It's not a lateral flow immunoassay, it's a little bit more sophisticated immunoassay. And immunoassays in hospitals, there's this so-called ELISA, the enzyme-linked immunosorbent assay. This is a classical clinical hospital method, and this is normally done in plastic microplates. But we wanted to have this on a more simple paper system. Why? because we can drastically reduce the sample volume. This is important if you want to work with blood, for example. We can drastically reduce the assay time. But normally when you move from these plastic plates, microwell plates, and the clinical instrument to paper, actually you lose sensitivity. So this has been done by us later on. And we use the CRISPR-Cas system here as an indicator to create a fluorescent signal. The fluorescent signal we can read out either with a lab instrument or with the smartphone. And here we targeted hepatitis B virus antigen. And just very shortly, how does this system work? So we use the CRISPR-Cas system to create a fluorescent signal. And for that purpose, we label an antibody with double-stranded DNA. And actually, the simple case would be to label it just with a few of these double-stranded DNA molecules. However, fortunately for us, we found this paper published in 2021, where a group showed a method how we can label an antibody, how we can attach many double-strand DNA molecules onto a single antibody. So please note, we do not want to detect DNA here. So we use any double-strand DNA, of course, that it should not be contained in blood, because otherwise the system would not work. And so because we add many of these double-stranded DNAs to one antibody, this increases the sensitivity of our system. Because now we combine it with the Cas enzyme. And in this case, when you have many DNA molecules on the antibody, we have many Cas enzymes, and that creates a very sensitive signal. So because time is really running out, so I will not explain too many of the detail again. So this is all paper here. Just work with paper and the reagents. So first we deposit the capture antibody. So this is an unmodified antibody that can catch the hepatitis B virus antigen. That's the first part. On another piece of paper, we put this detection antibody that we have labeled with many copies of double-stranded DNA. On a third piece of paper, we deposit the Cas12a CRISPR RNA complex and single-stranded DNA marked with a fluorescent dye, which serves as a signal creator. And in order to understand how this device operates, it's easiest when I show you a movie. Actually, here, this is a sped-up movie. So now this is a movie of the real device. And while I run the movie, you will see here on the right what is actually happening on the paper. So I said in the first step, the sample is added. Here, this is just water. When we add the sample, actually, we introduce our antigen. So in this way, we introduce our antigen. Then we do a folding, and now we add a buffer. And what does the buffer do? It simply washes away all unbound compounds. Next, we do a second step of folding, and we add another buffer. Now this buffer washes down this labeled antibody, and we form what is called the sandwich structure, sandwich complex. It's antibody, antigen, antibody. Now we do another folding step, and we add a further buffer, and that further buffer will now wash in the Cas enzyme. And this will activate the system and will start chopping up these pieces of DNA and create the fluorescent signal. And that's what we observe. So that's what we see, the fluorescence, if we look at it, for example, through a smartphone. And then we use simple color analysis. So we detect the green intensity, and we can make a calibration curve. On the x-axis is the concentration of the hepatitis B surface antigen. The y-axis shows the fluorescence intensity, now in this case measured with a laboratory instrument. So this is a very expensive, highly sensitive, cooled CCD camera detector. But I show you that we can do differently. But here I just show you, okay, this system works, of course, in water. it also works in blood plasma, and we get really, really low limits of detection. Just to let you know, the FDA, the Food and Drug Administration, requires a test that can detect this hepatitis B surface antigen at concentrations lower than 500 picogram per milliliter. So we are very well within that range. Just very shortly, we also detect the specificity, and as you can see here, so we only get the fluorescence for our target compound, whereas the fluorescence is not visible or hardly visible if we don't have our target. And as a next step, we wanted to show that we can also do this with blood. And so we did not do this with human blood because we did not have access in Japan to human blood samples of people infected with hepatitis B. So here we used pork blood and we spiked it with the antigen. So we spiked it with the hepatitis B virus antigen. And we introduced another piece of paper which removes the red color. It simply removes the red blood cells from our paper. And we can still get a very good limit of detection, about 210 picogram per milliliter with this system in whole blood. So you don't have to do any sample pre-treatment, just add a drop of blood to the device and look at the signal. And we also compared this with the commercially available system. So please note that this commercially available microplate system is run on an expensive instrument. It's run on a microplate reader and not simply with a piece of paper. And as you can see from this simple table here, first of all, we need a much lower sample volume. We are much faster. We can work at room temperature, and actually we have a lower detection limit than the commercially available system. But here still we use this cool CCD camera reader, and the final step that I'd like to show with the very strong support of Professor Papita Nath here. And so he helped us develop, or actually he constructed this simple smartphone adapter readout based on an LED light source, lenses, and a dichroic mirror integrated into first 3D printer, but later aluminum manufactured adapter to a smartphone. And so we have our paper device, we do our analytical test, and then we simply slide the devices through this device, and we look at the green color of the smartphone, and we use a standard app on the smartphone and to extract the color. And now, just to show you, like this compares the results obtained with smartphone and with the imager. Of course, the sensitivity of the smartphone is not as high as the cool CCD camera system, obviously, but we still receive very, very good good results with that. And this work is currently under revision at ACS Sensors, and so please keep your fingers crossed. I hope that we will have a successful review or comments on this. Okay, let me just summarize this part. So this has been one example of our CRISPR-Cas work that we are recently doing. We are doing also others, and yeah, these systems can be very rapid. Actually, they can be shorter than the commercialized systems. They can be shorter than the clinical, that the system that is used in clinical hospitals. They can even perform better. So that is a very interesting result, I think. And of course, in the system that I've shown here has been for hepatitis B, but if we choose a different capture antibody, we can also do this with other systems. Okay, before I finish, of course, first of all, I'd like to thank all of my collaborators that have helped with the work that I've shown today. So these are not all of my collaborators, including here Professor Papita Nath from Keio University, who stayed in my lab for 10 months. Then I'd like to shortly acknowledge, of course, all the students, or these are the students who did the actual work. So they did all the experience that I have shown you today. And as Papita knows very well, especially with Hikaru Suzuki, he worked on this hepatitis B sensor system. And finally, I'd like to thank all of my group members and very important also all of our sponsors. So here are our most important sponsors are mentioned on these slides. And thank you very much for listening to me. I'm a little bit over time and I hope that you found some interesting aspects. I know that most of you are working in physics, which is totally different from what I have presented. Yeah, but I hope that you have found some interesting points here. So thank you very much for your attention.
Thank you, Professor Daniel, for such an insightful talk. I wish that you could hear the round of applause from the audience because we are all online actually. Most of the audience are online today. So the session is now open to take some quick questions because we're already not already is 1 hour over.
Yes.
So we can take as a few quick questions from the audience.
Thank you, Professor Daniel. It's indeed, very interesting talk, and I'm really excited to see the results of your this very innovative way of detecting the fluorescence. The cleaving the Cas12 and then getting the fluorescence for the detection. It's really very interesting to know, and definitely would like to know from you more in this regard. But can you tell us about the mechanism of this fluorescence to have after cleaving it?
Yes, actually at this moment, do you still see my slides?
Yeah, we can see still.
Okay, let me see then if I go back. Yeah, first of all, I have to say that the Cas-induced cleavage, I mean, that's not a system that we have invented. So that is already used also in some clinical assays. We have adapted it to the paper. But what I would, as from the mechanism, so the process involved here is physically relatively simple. It is based on the so-called first resonance energy transfer. So we have, let me go. Yeah, well, okay, this is the movie, but yeah, this is a very schematic picture. So we have a piece of single-stranded DNA to which we have a fluorescent dye connected on one end and a non-fluorescent dye at the other end. So in this state, so when the single-strand DNA probe is in its original state, the fluorescent dye and this non-fluorescent dye, it's called a black hole quencher, are in close proximity. That means that the excited light creates the fluorescent dye in its excited state. However, instead of emitting light to return to the ground state, the energy is transferred to this quencher. So we don't observe a fluorescent signal. And so that's a system known as FRET, F-R-E-T. So I'm not sure whether you have heard about this. It's first resonance energy transfer. However, when the Cas enzyme is activated, as indicated by these small scissors, it will cut this, so it will separate this quencher from the fluorescent dye. And once they are no longer close to each other, this resonance energy transfer is no longer possible and the fluorescence appears. So that's the principle behind this fluorescence signal turn-on system. So I don't know, I hope this answers your question. But that's not something that we have invented. That's a very well-known phenomenon.
Thank you.
Yeah.
Professor Daniel, this is Mahanta. It was very informative talk and lot of information. And in just 1 hour, we cannot understand everything, but many things are really good to see that what is happening so far diagnostics and clinical applications are concerned. So I would like to just know one thing. What caught my attention is this biotennilated streptavidin, avidin, they give you very strong bond. And
Yes.
whereas you are sensing an analyte, maybe it was DNA like DNA or something else, I do not know. Can you come back to that slide, please, and please explain how this sense?
Yes,
Yes, yes,
yeah,
please.
yeah, yeah. Actually, yeah, here, so you are absolutely right. Yes, so streptavidin biotin is a very strong bond. And yeah, sorry, here I did not have enough time to explain that. So here, we simply use the streptavidin biotin combination to attach this DNA, double-stranded DNA, to our antibody. So we use an antibody which can bind the hepatitis B surface antigen, and actually that's commercially available. You can buy that, and you can also buy its biotinylated version. So here there's a biotin on that antibody. And if we combine, and we also buy the DNA which has a biotin on one of its ends. So by using streptavidin, it simply acts as a linker to connect this DNA to the antibody. And if we use just a single biotin on the antibody, actually one streptavidin can bind to 4 biotins. So of course, this is a schematic figure, but we would get, like in this case, one antibody with 3 of these DNA and this Cas system. So we have 3 activated Cas enzymes here. But here we use this doubly biotinylated DNA, and therefore such network structure can form. And the only change here is that we dramatically increase the number of these Cas enzymes on a single antibody. Of course, these are all schematic figures, but by doing so, we have a lot of enzyme on one antibody, and this gives us a very high cleavage activity, so that when finally, sorry, going through this, so when we are in this activated state, we can cut a lot of this probe, and therefore we get a very strong fluorescent signal. So the biotin avitin here is simply used as a linker to connect the DNA. The DNA in this case is not our analyte. So here we detect an antigen, so it's an immunoassay. That might have been a little bit confusing. So, but we just use the biotin and avidin to connect the DNA to our antibody.
So a single stranded and double stranded, both kinds of DNAs can be considered in this case, right?
Basically, yes. However, yeah, this is also another thing. So there are different types of these Cas enzymes, and we use the version called Cas12a. And Cas12a is most active for recognizing double-stranded DNA here. So that's why we use, for labeling our antibodies, we use double-stranded DNA. Actually, the Cas enzyme can also be activated by single-stranded DNA, but its activity will be much lower. So, but there are other types of Cas enzymes, but the Cas12a that we and many others use for such bioassays, they are activated by the presence of double-stranded DNA, and that's why in this case, when we label our antibody, so yeah, we use double-stranded DNA here.
Thank you so much.
Thank you. You're welcome.
Yeah, Professor Daniel, we have one audience, Professor Satyasundar Bhattacharya. He is online, so he has a question.
Yes, yes.
Yeah, Professor Satya.
Yes. It's a very interesting talk, Professor Daniel. Thank you very much for making it so easy to understand. Now, I have just my curiosity is that this paper analytical device that you have mainly presented, can that be also for chromatography? Can it be used for chromatography, like paper chromatography would do?
Yes, thank you very much for the question. Well, yes, the answer is definitely yes. And actually, I mean, let me show you one slide. Actually, we are doing kind of paper chromatography, but here we don't use it for separation purpose. So in these devices where we create this target concentration dependent difference in length of color changed paper sections. So actually this is paper chromatography. You can think of this as paper chromatography, but here we do not use it for separation. So we use it like in chromatography. So I guess you're familiar with chromatography, then you know about
Yes.
the RF value and also in thin layer chromatography. So of course, yes, here we use the fact that the RF value of this protein is lower than 1. That means the protein interacts with the paper surface. So this is, in other words, it's nothing else than paper chromatography, but we don't use it here for separation. We use it to retain and to deplete our analyte on the paper. And this we can do either with the unmodified paper for the protein will strongly stick to paper. So we use cellulosic filter paper. And in this case, for creatinine, we use a chemical reagent here, which will actually bind the creatinine. So it's not only the chromatographic effect. But of course, you could at the same time use these, and there are other people, now there are worldwide many people who work with these paper-based devices. Actually, also in India, there are quite many people. And yeah, I have seen people using these in combination with paper chromatography. So you can also do separation at the same time.
Okay. Thank you for your answer.
You're welcome.
Dr. Shivashish has some questions perhaps. He's online actually.
Thank you, sir. Professor Citterio, can you hear me?
Yes, I can hear you.
Actually, as you have discussed about some slope-based paper analytical device, then you have moved to QR code based, then again you have moved to this traffic light system based paper-based analytical system. I want to know that what are the shelf life of these systems that you have developed?
Okay, thank you very much for the question. So again, this is a very important question. As you have probably seen, so for the final ones here, actually, well, no, I've not shown. Actually, for the final one that I showed, this hepatitis B antigen, we have tested the shelf life. I removed the slide for time reasons, but this is not a full test. Yep. Here, this is for the hepatitis B surface antigen. So this CRISPR-Cas system, we have checked. In this case, we checked only for 1 month. So here, 1 month, this was stored at -20 degrees. So it must be frozen. So that's one point. We had to use a freezer. So this was 1 month. So in general, for this paper-based device, my answer to your question is it depends. So the system with the slope or the intercept that we use. So this is a pure chemical system. So what do I mean with pure chemical system? There are no enzymes, no antibodies involved. So yeah, it was this system that I showed you. So these are colorimetric indicators which are stable. So these devices, to be honest, we have not checked the shelf life here, but I am very sure. So this can be stored for a long time, even at room temperature. And for the traffic light system, I guess as well, we have not tested that. But yeah, the very important point is my answer to the question. I have been asked this question very often at conferences, and the answer is always it depends. So if you have more delicate molecules like antibodies or so involved, the shelf life might become shorter. And as if you have enzymes involved, it might become shorter, but there are ways of doing stabilization. And yeah, it has just been 28 days, but for the CRISPR-Cas system, so yeah, we tested that for 28 days and it was fine. But I cannot give you a general answer. So the more delicate the reagents are, the more delicate also devices will be. But yeah, let me just add, without being too long for my answer, just add shortly showing you, I think I also showed this slide. Actually, the left graph has been obtained on the paper surface and this with the solution. So the paper can actually have the paper with some stabilizing reagents that are involved here. So if you want to know more, I have to refer you to this publication. But actually the paper can be stabilizing. So if you look at the right graph here, this is in solution. Even after 1 week, you will see that the intensity of the signal here in the solution system stored in frozen state will strongly decrease, but the paper can have a stabilizing effect. Of course, again, this cannot be generalized. This depends on the situation.
Okay, thank you, Professor.
Thank you.
Yeah, we have. I think we can take only 1 or 2 questions for that. I think somebody is there who's raised. Chanika Setia, are you there? She raised her hand. Okay. No. All right. If not, if Chanika is available, no. No. Hello.
Yes, I am still here, but
Hello,
I
sir.
don't see anyone.
Sir, here in
Okay.
paper-based analytical device, you use paper as a substrate to make this kind of analytical device. I want to know,
Yes.
sir, can we use plastic and glass as a substrate to make this kind of analytical device?
Okay, thank you for the question. Well, the answer is basically yes, but, and here's quite a big but. If you have a flat glass or plastic surface, the surface area that you have available for immobilizing or any reagents is much, much lower compared to paper, because paper is a highly porous material. And of course, with plastic and glass, depending on the situation, you can create capillaries. You can work with glass capillaries that you modify. Well, another advantage of the paper that is very important for us is this the porosity, which helps us to have spontaneous liquid flow, which depending on how you design a glass or plastic-based device, this will not always be the case. So for many microfluidic devices made of plastic or glass, you will need a pump, you will need active pumping. But yeah, and the surface area that you have available for storage of reagents is definitely much higher on the paper. Well, not only paper, on any porous substrate. You could think, and there are people now using that. We actually have tried that, but there are also some reproducibility issues. I don't know if you are familiar with the process of electrospinning, where you create porous networks of artificial polymers. So paper actually is you can look at it as a polymer substrate. It's a biopolymer. It's cellulose. Cellulose is a biopolymer. And we have cellulose fibers which are randomly oriented and create such porous structures. So you could think of using not cellulose, but using the method of electrospinning. You can fabricate porous substrates made of polymers. Yeah, that would be an alternative, but it's definitely more costly. And but if you look and glass, sorry, yeah, of course, if you use a glass fiber porous substrate, yes, you can use. They are also partially used. The commercialized COVID-19 rapid antigen test has some glass fiber parts pieces in it, and you can use that. And yes. So in that case, the answer is yes, but I would still say you have the higher surface area for the paper substrate. But if it's a totally flat, a non-porous glass or polymer plastic material, you have a very, very comparatively low surface area to immobilize reagents.
Thank you, sir.
Hello, Professor Daniel.
Yes.
In the storage methods, we have used the desiccator. Is there any certain pressure level that has been maintained for the storage capability?
In the desiccator, no, the desiccator that we use is not a vacuum desiccator. So it's simply a cabinet. It's a plastic box which protects the devices from UV light and has a drying system. So it contains a desiccant like silica gel inside and just simply keeps the humidity at a very low level. But it's not under vacuum in that case.
Okay.
So it's atmospheric pressure.
Okay.
Okay, so Professor Daniel, so we are almost running out of time. Thank you for giving your valuable timing for this talk.
Yeah, thank you very much for giving me the opportunity. And
it is
yeah,
indeed
thank you
great
very much
pleasure.
for listening to my talk.
Yeah, in fact, we would like to have you in person here at Tejpur in future.
Of course, I hope definitely. I've never been to India. I definitely like to visit India. As I said, in India now there's a growing community. Actually, at the conference, I was at a conference, the MicroTAS, it's the Micro Total Analysis System conference just last week in Montreal, Canada. And there are quite a few people who present, of course, not only paper, but paper microfluidics, paper-based analytic devices. And I have also talked to some students presenting from India, presenting the poster, and they show very interesting work. So I know there's a growing community in India working on this. Yes.
Right. So yeah. So is there any plan to have this meeting or this conference here in India in future? I mean, about the microtasks.
No, actually not. The microtask is in rotation. It's organized by a society called CBMS, Chemical and Biological Microsystems Society. It's a non-profit organization located in the US, and they rotate between North America, Europe, and Asia.
Okay.
And yeah, well, of course, if there would be a person organizing that in India, but right now there's no person on the CBMS board from India. Actually, I'm also not on the CBMS board, but yeah, so I don't know about any plans to hold it in India. So I know next year it will be in Australia. So Australia, they count it as Asia in this case, yes. It has been in Asia. It has been in China, it has been in Taiwan, it has been in South Korea, and I think it has been in Singapore, I'm not sure. And it will be held in Australia next year.
All right. So thank you so much once again for giving your valuable timing for this talk.
Thank you very much for giving me the opportunity. It has been nice hearing or seeing, like you are very small on my screen. Yeah, but I still recognize
yeah,
your
we
face,
can actually zoom in the camera, but I don't have that switch with
no,
me.
it's fine. That's not fine.
Here
I see you. I see you. I see you
I
there.
can
Yeah.
see you sitting here. Next to me, Professor Pritam Dev. He is a professor of this department. He is working in the material science, and Professor Dambara Mahanta is again working in nanoscience and technology. And all are my students sitting in my back. They are from masters.
Okay, hello.
Some of them are PhD students, and of course, those who are in online, they are from other departments and other institutions. Okay, Professor Daniel, so perhaps we can log off now.
Okay, thank you very much.
Thank you. Yeah, we'll see you soon. Okay.
Thank you very much. Goodbye. Bye-bye. I will be leaving. Goodbye. Thank you.